So let's study glycolysis. Our first molecule is glucose, and then we have glucose 6-phosphate. So what I've done here was use a connection, and that connection is glucose to glucose 6-phosphate. Then after that we have fructose 6-phosphate to fructose 1,6-bisphosphate, and I use the fructose as our connector so that we can remember all of our molecules. From there it splits to dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. From glyceraldehyde 3-phosphate we have 1,3-bisphosphoglycerate. So my connectors here are phospho, we see that in both molecules, and glycer, and we see that in both molecules. You can also use the 3 as a connector. So from there, 1,3-bisphosphoglycerate, we have 3-phosphoglycerate, and we're moving that down. After that, we have 2-phosphoglycerate, we're moving that down. After that, we have phosphoenol pyruvate, and our connector is phospho. We know our next molecule after that is pyruvate, so it's phosphoenol pyruvate. What enzymes play a part in glycolysis? Well, we're going to study that next. So from glucose to glucose 6-phosphate, we have the enzyme hexokinase. Now we're going to look a little bit closer into that name, hexokinase. We're going to break it down. So the prefix hexa means 6, and that is a great way to remember the enzyme that is between glucose and glucose 6-phosphate. Now from glucose 6-phosphate to fructose 6-phosphate, we have phosphoglucose isomerase. Now our connectors are phospho for phosphate and glucose 6-phosphate. All you have to do is just remember isomerase. So it's phosphoglucose isomerase. Our next enzyme we have from fructose 6-phosphate to fructose 1,6-bisphosphate, we have phosphofructokinase. So our connector is phospho, because we see that in phosphate. And we also have fructose, and we see that in fructose 1,6-bisphosphate or fructose 6-phosphate and kinase. So that is our enzyme. From fructose 1,6-bisphosphate to our 2 molecules, we have the enzyme triose phosphate isomerase. I have the 3 written down because in the enzyme name, trio means 3. Now from glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate, we have dehydrogenase. Now from 1,3-bisphosphoglycerate to 3-phosphoglycerate, we have kinase. From 3-phosphoglycerate to 2-phosphoglycerate, we have mutase. And then from 2-phosphoglycerate to phosphoenol pyruvate, we have enolase. From phosphoenol pyruvate to pyruvate, we have the enzyme pyruvate kinase, and our connector is pyruvate. Energy is created and used, so we're going to explore these reactions. So from glucose to glucose 6-phosphate, energy is used. From ATP, our energy, to ADP. Between fructose 6-phosphate and fructose 1,6-bisphosphate, energy is needed. So we see ATP jumping in, giving us the energy that we need for this reaction. Dihydroxyacetone phosphate can convert to glyceraldehyde 3-phosphate, and it can happen in reverse as well. Glyceraldehyde 3-phosphate is then oxidized, transferring 2 hydrogen atoms to NAD+ to form NADH. From 1,3-bisphosphoglycerate to 3-phosphoglycerate, ATP is created. More ATP is created from the high-energy phosphate group from phosphoenol pyruvate. So with the Kreb cycle, I created a key, and these are the things that I want you to remember. CS and Fumalo. I know it sounds funny, but we're going to start with CS. So around the circle, we're going to write C, I, A, S, and there is another S. And then we're going to do Fumalo. We have Fu, Mal, and O. So we're halfway there remembering our molecules. So our C is citrate. Our I is isocitrate. A is alpha ketoglutarate. Our first S is succinyl-CoA. Our next S is succinate. Then on to Fumalo. So we have Fu, which is fumarate. And then Mal is malate. And our O, oxaloacetate. So on to our enzymes. We have in green the dehydrogenase. So the first one between pyruvate and acetyl-CoA, we have pyruvate dehydrogenase complex. Between isocitrate and alpha ketoglutarate, we have isocitrate dehydrogenase. Alpha ketoglutarate and succinyl-CoA, we have alpha ketoglutarate dehydrogenase. So from succinate to fumarate, we have the enzyme succinate hydrogenase. And then from malate to oxaloacetate, we have malate dehydrogenase. The next two enzymes are synthases. The first one is between succinyl-CoA and succinate. This enzyme is called succinyl-CoA synthase. The next synthase is citrate synthase, and it's between oxaloacetate and citrate. We have our two last enzymes. The first one is aconitase, and it's between citrate and isocitrate. And our next one, our last one, is between fumarate and malate, and it's fumarase. Now from pyruvate to acetyl-CoA, carbon dioxide is released, as well as NADH. Now isocitrate is oxidized. It gives us NADH from NAD+. Now decarboxylation comes into play and carbon dioxide is released from the cycle. Now we see the same set of reactions happening between alpha ketoglutarate and succinyl-CoA. Between succinyl-CoA and succinate, energy is produced by substrate-level phosphorylation. Now succinate is oxidized to produce FADH2 from FAD. And malate is oxidized to produce NADH from NAD+.